Hello, everyone, and thank you for joining us today. My name is Andy Helminger, and I'll be your host for this NASA Technology Transfer Program webinar on NASA's method for rapid detection of trichloroethylene, or TCE. Our presenter today is Amy Keith. Amy holds a bachelor's degree in electrical engineering and a master's in environmental engineering. She began her NASA career working in instrumentation at Marshall Space Flight Center's wind tunnel. She later worked at Stennis Space Center as a test engineer for the Space Shuttle main engine, and then served at Stennis in the External Tank Business Office and Advanced Turbopump Program Office. She currently works as an environmental engineer in the Office of Center Operations at Marshall Space Flight Center. Several years ago, Amy gathered a team of biology and remote sensing experts from the State University of New York and the Naval Research Laboratory. This team set out to fulfill a vision they had of developing an alternative to current methods for detecting contamination in the environment. These current methods are often expensive and slow. Amy and her team have isolated light wavelengths that correspond to a plant's exposure to trichloroethylene, or TCE, and have been working towards creating an inexpensive, non-destructive, and rapid method for detecting TCE, as well as other chemical pollutants in contaminated areas. Following Amy's presentation on this technology, Jordan Larson, a technology transfer specialist at Marshall Space Flight Center, will be giving a short presentation on how NASA licenses technology to outside organizations. Before we get started, I'd like to point out that your microphones will be muted throughout the presentation. So if you do have any questions, please type them into the chat box in the lower right corner of your screen, and we'll answer them during the Q&A session at the end. So at this point, I'm going to turn it over to you, Amy.
Hello, everyone. So glad that you joined, and I'm really excited to share this idea with you. Next slide. These are the partners that Andy mentioned. I have a biologist and a graduate student from the State University of New York helping us with this project, and also we have a remote sensing expert from the Naval Research Laboratory helping out. Next slide. How all of this got started was I was reading an article in our local Marshall newsletter about a group of scientists who were doing some experiments with hyperspectral imaging, and they were using it to detect the differences between natural corn and hybrid corn. And this is just a scan of a cornfield, and you can see how the different types of corn come out as different colors in the scan. So next slide. So looking at this idea, I thought, well, if they can use this technology to tell the difference between hybrid corn and a natural corn, is it possible that we could use hyperspectral imaging to detect the difference in plants that had been exposed to contamination and plants that had not been exposed to contamination? So what is hyperspectral imaging? Everything that you see because it reflects light. And the way the light is reflected from an object is characteristic of that object, comes from the characteristics of that object. So if the characteristics of that object are changed, then the way the light is reflected from that object change. And hyperspectral imaging can measure those changes in the way the light is reflected, or spectral signatures. Next slide. So in the environmental world, trichloroethylene is one of the most common contaminants. So we decided to start our experiments with trichloroethylene. It is across several of the NASA centers because we used it to clean our rocket parts. We would flush the engine, specifically the nozzle of the engine, because it would be covered with soot. To clean it out, it would flow down the test stand and into the groundwater. And you can see it also had other uses, such as dry cleaning. That was a big one. So we started out with trichloroethylene. Next slide. And this is just a description of what we were just talking about. The photograph or the schematic on the left shows that you have TCE, it pulls on the ground and eventually works its way through the soil and into the groundwater. And it is the most common contaminant in Superfund sites across the United States. And the drawing to the right is Marshall Space Flight Center, and all the yellow are groundwater plumes that are contaminated with TCE. Next slide. So how would we go about figuring out how to use this technology to detect trichloroethylene in the groundwater via plants? So we know that plants take up TCE. As a matter of fact, plants that take up TCE in our laboratory experiments are actually greener and healthier than the plants that we don't expose to TCE. So if we could measure the way that the plant spectral signature changes when it is exposed to TCE, if we could isolate the wavelengths, the spectra that change when the plant is exposed to TCE, then we could invent something that maybe could clip onto a leaf, you could take out into the field and see if a plant had been exposed to TCE, and then know if your environment had been exposed to TCE. Next slide. So what we did is we came up with, we're using a handheld sensor now. We clip it onto the plant. It gathers spectra like you see in the middle there, and then those spectra are downloaded to a laptop or a tablet, and then we take those and we analyze those. Next slide. So what we wanted to do with this data that we're collecting, we wanted to identify if there were key wavelengths where exposure to TCE can be recognized. Are there wavelengths that we see changes in from a clean plant versus a dirty plant? We needed to see if we could isolate changes due to TCE exposure from other naturally occurring stresses, such as nutrient stress and water stress. And we wanted to make it something that you could take into the field and easily carry into the field, clip on a leaf, and get a reading. Next slide. So we started out with some investigations. We did some experiments. We grew some poplar trees in a greenhouse, and we nutrient stressed them, water stressed them, and exposed them to TCE. Next slide. So you can see this is our greenhouse where we grew our plants. And in our initial investigations, we used a large desktop hyperspectral imager because it gathers a lot more spectra than a smaller radiometer or a handheld spectrometer. And we wanted to gather as many spectra as we could to have a better chance of identifying a wavelength that would change when exposed to TCE. Then we gathered all of those spectra and put them into a reference library. Next slide. Then how we collected these spectra is on each of the leaves, we decided that we would choose pixels from 2 sets of regions of interest. The first set of pixels we used to train our classification routines, which were statistical methods. And we said, okay, statistical method, if you see this sort of wavelength, then this is a wavelength from a contaminated plant. Classification routine, if you see this sort of wavelength, it's from a clean plant. Classification routine, if you see this kind of wavelength, it's from a nutrient or water stressed. And then we use those libraries, those classification routines, and bounced those against scans of leaves that we knew were TC exposed, nutrient exposed, to see how accurate our classification, our statistical method was. Next slide. So when we did this, when we took our scans of our leaves, compared them to our spectral libraries, we got about an 80% accuracy of correctly identifying when a plant was exposed to TCE. Next slide. So we decided to do another experiment to try to refine our results a little bit. The first experiment was we wanted to know, when is TCE detected? How long does a plant have to be exposed to TCE before we see any changes in its spectral signature? Then the second experiment was we wanted to know how low could we detect TCE? At what level did we stop seeing the changes in the spectra? Next slide. So here's our experiment. We took TCE and dosed plants at the same dosage for 1, 2, 4, and 10 weeks. And then to determine how low we could see TCE, we dosed them for 10 weeks at 5, 50, 100, 1 ppm, and 100 ppm. Next slide. And here's our results. We began to see that accuracy started to fall off on our TCE detection between 50 ppb and 100 ppb. Next slide. And then we began to see that we could start to see a significant change in the spectral signature when exposed to TCE after the plants had been exposed to TCE for about 4 weeks. Next slide. So just to summarize everything we've found up to this point, we're getting around an 80 to 85% accuracy in detecting TCE. We did some other experiments where we removed leaves and delayed scanning them for up to 15 minutes to see if that made any difference in the accuracy. We saw no difference there. We also looked at leaves from different parts of the plant, the younger leaves at the bottom and the older leaves at the top, to see if that had a change in the accuracy. We saw no change in the accuracy there. And then, as we talked about earlier, we wanted to see if we could detect stress, nutrient, and water stress plants, differentiate that from the TCE, and we were able to do that with an 86 to 99% accuracy. We were able to detect it after 4 weeks, and we refined it down where from between 50 and 100, we figured out that 85 ppb was our lowest limit that we could detect TCE. Next slide. So then we started thinking about how we could apply this. We were thinking springs and seeps have a lot of natural vegetation around them. So we were thinking that would be a good place to measure groundwater, because a lot of groundwater daylights in springs and seeps. Wetlands, it would be a good way to measure the health of wetlands. And water discharges like that are required by permitting. You could use this to measure, if TCE was on your permit, you could use this to detect TCE. There are also systems that we use to clean up contamination. You actually plant plants in a contaminated area. They take up the contamination and metabolize it. And usually you have to destroy that system. This would be a non-destructive way. You could go in and clip on the leaf, see if your system was working or not, without having to take core samples or leaf samples to determine if you were taking up the contamination. And you could use it to delineate contamination. If you had a span of trees over an area, you could use it to determine where one tree had contamination and another didn't, to kind of quantify an area of groundwater contamination. Other applications would be real estate transfers, if you just wanted a quantitative evaluation of if contamination was present on a site. And then for farming, to help farmers keep up with the health and potential contamination of their crops. Next slide. So this is just a summary of some of the benefits. Right now, if you want to go out and investigate, if you have contamination in your groundwater, you have to put a well in. It's expensive, and you have to send samples off to the lab, which usually takes a couple of weeks. And it's non-destructive, as we were just talking about. Next slide. So this method is available for licensing, and that is all that I have. Thank you so much for listening.
All right, thank you, Amy. We've received a number of questions during your presentation, and we'll get to them shortly. First, Jordan Larson is going to give us a brief overview on how businesses can partner with NASA through technology licenses.
Thanks, Sandy. Amy, that was a wonderful presentation. This technology, with 1,400 others, can be viewed in our patent portfolio, which can be found at technology.nasa.gov. We went ahead and arranged everything into 15 different categories where you can browse stuff in health and medicine, materials and coatings, propulsion, sensors, optics, and more. If you're interested in looking for something a little bit more specific, you can search by keyword. NASA's Technology Transfer Office also gives away our software that we've developed in-house for free. Currently, right now, there's around 1,000 different software codes where you can see our software catalog at software.nasa.gov. That website is set up primarily the same way, where you can search by category or by keyword. Next slide. Once you find a technology on our website, technology.nasa.gov, you will see a page that looks like this. Here at NASA, we call this a TOPS page, and it's where you can read a little bit of information about the technology, the benefits and why we had to develop it, and different applications that we think it could be useful for. Down at the bottom of the page in the black box, there is contact information where you can get in touch with our licensing concierge, and he can arrange a meeting where you can talk to the inventor and learn a little bit more. Next slide, please. We have 3 different licenses available at NASA. We have commercial, in both exclusive and non-exclusive, evaluation, which is a flat $2,500 and lasts 1 year. It's a great resource to test drive the technology before committing to a commercial license. And we also have a startup license, where we waive all upfront costs for the first 3 years. And after that, or when you start selling a product commercially, there's a small running royalty associated with the license. Licensing a technology is quite easy. You just go to that website that I mentioned before, technology.nasa.gov, and click on the technology you're interested in. There's a big button on the screen that'll say apply now, which you will click, fill out an application, and we will contact you and set up a meeting where you can talk to the inventor, see if it's a good fit, and from there you can negotiate the terms of the license. And that's about it. Next slide.
Great. Thanks, Jordan. Thank you, Amy, and thank you to all those who attended today and asked some great questions. If we didn't get to your question during the Q&A session, we'll follow up with you by email. If you have additional questions, please feel free to submit them to the email address at the very bottom of the screen, and we'll make sure to follow up with answers to those as well. Once again, we really appreciate you taking the time to be with us for this presentation. We hope everyone has a great day.